I have two things to say. I'm very grateful for the introduction Jack gave, and I'm very grateful that Koji Nakanishi did not do it. If any of you ever heard Koji's introductions, you'll understand why. I don't want to go through that, but they're really wild. But Jack gave a very nice civilized introduction with the exception of that one inorganic piece that he stuck in the middle there. And I'm not sure what that was all about. Anyway, let me tell you about this, because this is a meeting devoted to some extent to chemistry and health. And so I want to tell you about an adventure we've had in this. We're going to talk about differentiation therapy for the treatment of cancer, because that's how we started, although you see it becomes a bigger story than that. And in particular, we found a number of compounds that we developed that could perform this kind of therapy. We tell how it worked, and then we'll eventually show you about their activity in vitro and then in vivo. And in vivo will include, of course, animals, and then eventually includes people. We're going to come to a compound that has been approved now by the Food and Drug Administration for treatment of cancer. It was approved in October 2006, so it's really been around for a while now and is doing extremely well from everything I've been able to find out. You'll see why I don't get direct information. Stem cells have two functions. First of all, they multiply and proliferate to make more stem cells. That's what cancer cells do too, of course. But then the stem cells also differentiate to form adult tissue cells with specialized functions. You know, a pre-erythrocyte turns into an erythrocyte, for instance. But that doesn't happen with cancer cells. They don't have that differentiation possibility, and that may be a flaw that one can overcome, as you'll see. If they don't differentiate, the result of this can be a form of cancer. It may not be the universal story about cancer, but certainly it's the story about at least one, which I'm going to tell you about now. Pre-erythrocyte is a stem cell that eventually turns into erythrocytes, which is to say red blood cells. But they're very different. This is spherical. The erythrocyte is flat, like a disc, essentially, like a frisbee. This has a nucleus, this does not, so once that happens, it can't go back, of course. And this is loaded with hemoglobin, and there's no hemoglobin in here. And so that's the normal thing that the stem cell does. It multiplies and it also differentiates. When it's infected with the murine erythroleukemia virus, murine means mouse. These are mouse cells that were studied by, you'll see by who did, I'll tell you in a minute who did it. The mouse cells were being studied, and a form of erythroleukemia was found in these mice if this virus had infected these pre-erythrocytes so that they no longer could differentiate. They still proliferated, they still multiplied as did the virus. And so one got these non-functional, spherical, no hemoglobin cells down here instead of the red cells. And so that's, of course, a disease that it's not the same as human leukemia, but you'll see that it is obviously something that one can talk about trying to deal with. Now, it turns out a woman named Charlotte Friend was studying these. These are sometimes called Friend cells because this particular group of cells is something that she pioneered. And what she was doing was soaking it in aqueous DMSO in order to soften it up so that she could transfect it, so she could get larger molecules into the cell. But when she did this with this rather high concentration of DMSO, what she found is that about 2/3 of the cells turned into red blood cells, and that had never happened before. And they had the form of red blood cells, and they were loaded with hemoglobin. They had no nuclei. They had actually undergone this transformation, which stem cells do, and which cancer cells normally don't. And so she came to my colleague at that time. He was the dean of the medical school. We'll get back to him later. Like I'd say right now, his name is Paul Marks. He was the dean of the medical school, and Paul knew about this work and came to me to talk about it, and he said fundamentally in so many words, what can we do about this finding? Here is Charlotte Friend's publication. This is Charlotte Friend's publication here. This is what happens if you don't treat these cells with DMSO. You can see they're white. There's a little bit of pink there, but there's not a serious amount of high hemoglobin in this thing. There certainly aren't differentiated cells here. But if instead it was treated with this DMSO, this added DMSO, then you in fact got these red cells, or erythrocytes. They had transformed, both the shape and the biology. I mean, everything had transformed. Now, one obvious requirement, you would think, well, this is great, a cancer cell turning into a normal cell, let's just do that to people. But of course, 280 millimolar DMSO is a pretty heavy load to think of putting onto people, and nobody, as far as I know, has done it. Our obvious reaction is, let's see if we can't make something that's at least more potent, right? And so we don't have to go to that preposterous level, but maybe that would still do the same thing. Now, this is what we found. We looked at whether DMSO was special in terms of its chemistry, and the answer was no. It's simply a polar sort of a compound, a little bit of hydrocarbon here and a polar group here, and that was the thing that required 280 millimolar DMSO. So we said, well, obviously carbonyl compounds, amides in particular, have that same structure. And amide, as you know, is a lot of N plus O minus to it, and so really you have that same sort of a dipolar structure here. We tried that, and it worked at 50 millimolar. For the medicinal chemists in the audience, let me tell you these numbers. This is the optimum concentration required for the differentiation. As I say, it did about 2/3 of the cells. This is the optimum concentration of N-methylacetamide. As you eventually know, we'll get to when we start talking seriously here, we're going to get to ED50s, because as you know, there's no such thing as defining the 100% number. Is it 99? Is it 99.9? Is it 99.99? You can keep going up and up. So that's not a defined number, but 50% is a defined number. So what people generally do in the medicinal business is talk about the concentration needed for a 50% effect. And so we'll do some of that. That's not these numbers. Okay, so the question is, how can we make it better? Well, after a lot of playing around with solvents, we said, well, we're really still in that region of 50 millimolar or so. That's not very exciting. But maybe if we double it up to take advantage of chelate binding, we might be able to get a more potent compound. And I explained this to my postdoc, a very, very smart, very nifty guy who's now running his own pharmaceutical company somewhere in California. But anyhow, I explained it to my postdoc, and I said, we got to just link these two amides together and see if chelate binding might give us more potency. You know, if this one binds and that one binds into a nearby site, then the combination will be better than if you had to saturate both sites with separate molecules, right? And so we said, let's do that. And he said to me, well, that's very amusing, but we have no idea that there are two sites. We have no idea how far apart they are, and therefore we have no idea what kind of a linker should we put between these two things. And I said to him, well, at least at the beginning, every chemist knows the answer to that. In the absence of other information, the magic number in chemistry is 6, right? So six methylenes in there, let's see what happens. It turned out to be the best. It's ridiculous. There's a rule called Murphy's rule. This is an anti-Murphy finding, right? In other words, everything that can go wrong will go wrong. The first guess was the best compound. There's a curve, 5, 6, 7. I didn't bring you all that stuff, but it's all published. But there's 5, 6, 7. 6 was at the top. And so that's great. Apparently there's some kind of a spacer here that we need. And you can also take this amide and turn it around, reverse it, and it still comes in at about 5 millimolar, again, for sort of optimum differentiation. Now, how do you make it better? Because we use that compound. This compound here, HMBA, has an acronym, hexamethylene bisacetamide. Believe it or not, that compound appears in a book put out by the National Cancer Institute called Generally Recognized Effective Compounds in the Treatment of Cancer. And that compound is there. It looks like the solvent from which most of the others were recrystallized, right? I mean, it's ridiculous little compound. It's still there. I was just at the NCI recently, and they showed me the book. It's still in that book. But obviously, that's nowhere near potent enough. And it wasn't. People who took that, we had one case of a woman with non-small cell lung cancer, cigarette smoker's cancer. And she had recovered with this, and after a year, her cancer was gone. And, but most other people couldn't tolerate these levels. These are very high levels, and most people couldn't tolerate these levels. And so we had to get more potency in order to get away from the side effects that we were getting with that. And so the question is, how do you make it more potent? Well, look, what can an amide do? It either binds to metal ions or makes hydrogen bonds. What else can you do with an amide group? And either way, a hydroxamic acid could be better, because it's a better metal ligand and it has more hydrogen bonding possibilities. And so if that's what's involved, let's make it. So let's make a bis hydroxamic acid. So we did that. That was easy enough. And we made this compound here, suberyl analide. This is suberyl bis hydroxamic acid. This is suberic acid here, the bis hydroxamic acid. And that got us down to 30 micromolar. All right, it's a start. Not a bad thing. And it's a little high still, and that again is the number that you need for maximum induction of this cellular transformation we were talking about. But that was not bad, and this went into some patients, but again, it wasn't ideal. And so then the question is, how can we make it better? Well, I said, we don't really know that we need a hydroxamic at both ends. Suppose this is binding to a metal ion, as it turns out it is. And then at the other end, you don't have any such metal ion over here. You just have some binding cavity of a more normal sort. Maybe we can get some hydrophobic binding by replacing that hydroxyl over there with a phenyl ring or some other hydrocarbon ring. So let's do that. Easy stuff, right? Just do it and let's see how it works. So we did that, and we went down to 2.5 micromolar. And this compound we call SAHA. It's sabaric acid, oil, anilide, hydroxamic acid, SAHA. And at this point, we had potency enough to take it seriously. This was what we had for treatment of the cells. What we found, we eventually got the binding constant of the target. And we identified the target. And when we did that, we were able to measure the binding constant. It's about 30 nanomolar. So it's really more strongly bound than this number would indicate. But when you give it to a solution of cells, you have to worry about permeability. You have to worry about the extent to which maybe other things happen. Whatever it is, you had to put more in when the cells were in suspension than you did when you actually finally identified the target. As I say, the 30 nanomolar is the binding constant, so that corresponds to a 50% inhibition in any case. So if you took this one down to 50%, maybe it'd be down to 1 micromolar or whatever. And you could again turn it around. Now, what happened then is that we looked at other drugs which had these general sorts of structures, and we had a number of things that were similar in the low single-digit micromolar region. And then this is a material called trichostatin A. It's a naturally occurring compound that's used as an antifungal, and it's about 30 times more potent than SAHA is. And you can see it's got a hydroxamic acid at one end, it's got a benzene ring at the other end with some other stuff. We put all this kind of stuff into our compounds. We made about 1,000 compounds one way or another around this general area. But SAHA was about number 300. SAHA was the number 300 of the ones we made. And we decided to take it and see what we could do with it. In other words, you had a compound that was working, let's run it through until we see what the problems are, and then we'll solve them by making a better compound. So we did that, and we just ran SAHA through to see what would happen. I should tell you that SAHA, that silly little structure I showed you, is the compound that was in fact now made it all the way through clinical trials, improved by the FDA, and is now in a very heavy use actually. So a little simple number 300. The people in the pharmaceutical business will wince at the idea the 300th compound you made actually turns out to be a drug, because they're usually in the thousands, 10,000, sometimes even more. There used to be 50,000 compounds in order to get one that works. It's now more like 100,000, so 250 is just ridiculous, right? Anyhow, here we are with this compound, and it worked against a lot of other cancer types. That's the first thing we did, is look at other cells. So we sent this thing down to the Food and Drug Administration, FDI, and they looked at the ability of this stuff to work with other cancer cells. They had a library of about 60 different kinds of human cancer, so they looked at all of them. And what they found is that we differentiate human colon cancer and adult HL60 cancers. This is regular leukemia that we have. This actually was done by my collaborator at Sloan Kettering, which I'll get to in a second. At the National Cancer Institute, they tested these compounds, this and a few other compounds related to it, and we saw that they gave differentiation, and they did three things. They caused cytostasis. The cells stopped growing. All of them stopped. That is, every cancer type stopped with this stuff, with a somewhat variable amount of compound needed, but everyone stopped growing. Then we got either differentiation, which is the thing we started with, or apoptosis, and about 50-50. At about half the time, the cells went into this programmed cell death, but it's something that is generally expected to be true of cancer cells and not of normal cells. There's an awful lot, there are many, many cases in which apoptosis can be induced in cancer cells, not in normal cells. We've looked at this. We have not seen in normal cells any evidence for apoptosis induced by this at the same levels at which we're getting the thing with the cancer cells. So it's not a unique finding, but it's a very good one. And it turned out it worked in all human cancer cells that they had. This is still more or less the battery they have, and they tell me that it's still true, that it does all the things which I've described here. Now, then we started looking at animals. In immunocompromised mice, we found that we could stop the growth of xenografts of human prostate and neuroblastoma cancers, and I'll show you that in a little bit. I'll show you one of the cases. Now, here's just a little bit of the kind of thing that we saw. These are the ED50s now, 50% cell growth, inhibiting 50% cell growth. And here we are with SAHA. You can see with this is human leukemia, HL60 leukemia, about 450 nanomolar needed, half micromolar. Non-small cell lung cancer, this is a cigarette smoker's cancer, and still it was, sub micromolar. Colon cancer, almost micromolar. This is neuroblastoma, it's a little over micromolar. Melanoma, a nasty childhood disease among other places. Ovarian cancer, prostate cancer. It was working with, and it goes on and on. This is just a sampling of them. Here's a compound we also looked at. It was somewhat more soluble in water because we put a pyridine ring instead of a benzene ring, and it turns out to be not as potent. And that, I think, reflects the fact that this is giving you hydrophobic binding, and this is simply less hydrophobic. So there's no special thing about the nitrogen. It just makes it more soluble and, at the same time, less likely to bind into hydrocarbon regions. Okay, I think one thing I will mention now before I go on, because I may forget to say it otherwise. Here's SAW up here with these 6 methylenes in it. And you say, well, what about that? Did we look at that at all? Of course we did. And it turned out 6 was the magic number again. 6 was the same thing. You needed 6 methylenes or the equivalent in length. You could have benzene ring that take part of the space, but you needed that kind of spacing of 6 methylenes. We said, well, where's the surprise there? That was true with the bis-hamides. It must be also true with the bis-hydroxamic acids or this monohydroxamic acid monoamide thing. Why not? And the answer is it isn't. Or putting it another way, the target for this class of compounds, the hydroxamic acids, we have identified, which I'll come to. The target is not the target for the bis-hamides. Nature, just to keep herself amused, decided to have the same optimal length of a class of compounds that were hitting two different structures. But we think we understand the reason for this 6 methylene, and you'll see why that is when we get to the way these things work. All right. So what was the effect of SAHA on transformed cell lines? This really just means cancer cells, okay? Erythroleukemia cells, we got differentiation, that's what I mentioned. Bladder carcinoma, differentiation, well, that was interesting. Neuroblastoma and myeloma, besides getting cytostasis, they went into apoptotic death, apoptosis. And human breast and human prostate, this is nice. They, in fact, did differentiation, but under some conditions, they also induced apoptosis. So you can see where we had sort of an equal opportunity lecture here, right? We're hitting everything. You're going to hear a little bit about breast adenocarcinoma if you go tomorrow to the physical chemistry lecture being given by Laura Kaufman. Laura Kaufman is one of my colleagues at Columbia, and she has been looking at this compound and its ability to change glioblastoma cells' ability to proliferate through a matrix. Glioblastoma is a particularly nasty disease because it doesn't stay localized. It sends out essentially tendrils and sort of moves through the brain, and it's almost impossible to get it all out without causing serious destruction. SAHA turns out to inhibit that, and she's going to talk about that tomorrow. It has nothing to do with me. We just gave her the compound. Okay. Here's the breast cancer cells. These are human breast cancer cells. And originally, they were spread out about the way they are here in a medium, but in fact, on a surface. And then when they were allowed to grow, a little bit of DMSO was present, but that's not relevant here. That's just part of that was part of what we used to help deliver that as well. But the DMSO itself isn't doing anything much here. And all this purple here is the DNA, that is the cellular nuclei. And in addition, this green stuff here is the milk fat. So milk fat is being produced by these cancer cells, but the red, if it were here, would be in fact the milk protein, and there really isn't any. So these cancer cells, these breast cancer cells proliferating, they fill the whole space. Nothing stops them. In addition to that, they do not have the adult function. They don't make breast milk. But if they are instead treated with SAHA, at a significant concentration here, 5 micromolar, so well over the ED50 here. When they're treated with that, you can see that what happens, they don't spread out all over the place. They stay in place. They make breast milk. Here's the breast milk protein. Here's the breast milk fat. These are normal breast cells. And so that transforms from that to that. That's, again, like what we saw originally with these erythrocytes. Now, how does this stuff work? At the time we were publishing this work, and at the time we were doing it, a man named Yoshida in Japan, a biochemist, biologist, but there's a number, it's a common name, and there's a well-known organic chemist named Zenichi Yoshida. This is not Zenichi, this is Yoshida, who the biologist there. He was studying a group of compounds. Trichostatin A was one of them, one I mentioned before, that's 30 times more potent than ours. 30 times more potent, but more toxic. And we'll come to that. The potency and toxicity are related in ways that turn out to matter. But here's another thing. Tripoxin B is another compound that also, in fact, introduces differentiation. Both of these induce cytodifferentiation in just the cells themselves. Putting them in animals is another matter, or people. This one turns out to be inhibitors of an enzyme called histone deacetylase, and he discovered this. So full credit to Yoshida, because we were trying to figure out what the target was, and he published that this stuff is it. In the meantime, we had been looking at what was the target here, but you can see all you have to do is look at SAHA and look at trichostatin A, and you would not be surprised to think that they might have the same biological target. And it turns out this stuff inhibits histone deacetylase, and so the question is, did our stuff do that? And of course, we looked at it. We had, in the meantime, been making a photoaffinity label. What you do in this is this was being done by, actually, believe it or not, this compound was made by an undergraduate in my lab, but then the tritium part was done by one of my graduate students. But what you do here is you irradiate that stuff in solution, makes a nitrine. That nitrine will let it bite into any protein it hits, so that's not terribly interesting. What you want to know is selective binding. That is, some selectivity. If you put the normal drug in, you can suppress the targets that this hits by binding into the same site, but you won't suppress just random attacks. And so this then differentiates and lets you know which are the ones that are having to do with the actual target binding site itself. Anyhow, histone deacetylase, obviously a great candidate, and we saw this exactly right. We published this now, I think it's 1998. Okay, 10 years ago already. Histone deacetylase was a target. The compounds inhibited the enzyme. The enzymatic activity ran parallel to the cellular activity. This was really quite important. We had, as I say, hundreds of compounds which were showing various kinds of degrees of activity. And what we saw is the cells, how much you had to put in to make the cells react, ran parallel to how much you needed to put in order to inhibit the enzyme. So at least that was a correlation. A correlation and a cause are not necessarily the same, but it certainly was strong evidence that we are on the right track with our stuff. And in addition, we got this specific labeling. That was one of the compounds that was specifically labeled in the sense that you labeled it, and if you put in the cold drug, you could suppress that labeling. So this was binding into the enzyme active site, and finally that azido compound was still an active enzyme, active compound inhibiting the enzyme, so that azid did not interfere with it. Okay, what comes after sequencing the human? Let me see if maybe I skipped it. Oh, here. We skipped this. All right, this is fine. Histone deacetylase, what's the story with that? This has to do with what comes after sequencing the human genome. We all know how important this is. There's great excitement about it, and it's almost complete. But once you know it, or even before you know it, these other things are what you need to learn. You need to learn what is the natural control of gene expression. What turns genes on and off? If all the genes were being expressed at full rate all the time, life would not be possible, right? Life requires not only an accelerator, but also a brake, right? You can't just go flat out with every possible gene. And many of the genes are not, in fact, expressed, or they're expressed only, say, in a particular part of life, maybe early parts of life or things of that sort. So what's the natural control of gene expression? And then how can you control it with small molecules? And that's the medicinal chemistry part. That's the part that chemists can do, and that's the part that we're involved in. Histone deacetylase is part of that control. Histone deacetylase helps regulate gene transcription because in chromatin, the DNA and histone combination, DNA wrapped around a bundle of proteins, histones, there are 4 histones in the core, 2 copies of each. So you've got 8 proteins in the core, and the DNA is wrapped around it. And the chromatin has the DNA, polyanion, wrapped around the histone, which is polycation. It's got a lot of lysines in it. So you've got a polycation core and a polyanion wrapped around it. That's chromatin. Histone transacetylase takes acetyl-CoA and uses it to acetylate some of the lysines. It helps to neutralize some charge, so that loosens things up a bit. But more to the point, it also promotes transcription because there's a code which has now been essentially derived and devised by David Alice, who was at the University of Virginia, but is now actually at Rockefeller University. And what David Alice showed is that there are particular lysines whose acetylation turns on the transcription of particular genes. And so that is really not a general on and off, it's a specific on and off. Really quite an interesting thing, the so-called histone code. And this has caused a lot of excitement, and of course it makes a lot of sense for us, because as you'll see, we turn on some genes, but not all. Okay, histone transacetylase promotes transcription, acetylating these things. Histone deacetylase neutralizes this thing neutralizes the charge when you put the acetyl groups on. Histone deacetylase takes them off again, getting rid of this code, the acetylated lysines, which code for, let's put this transcription complex together. And in addition to that, of course, it puts the charge back on, and it suppresses transcription. We block this, so we upregulate that. So what we do, at least in the simplest picture, which I'm giving you now, simplest picture only, we actually turn on gene transcription of particular genes by blocking this deacetylation. Now it turns out, interestingly, nature is much more interesting than simple ideas. And about half the genes, we've looked at gene chips, about half the genes that we modify are turned on, and another half are turned off. So forget everything I just told you, right? But in fact, what it really means is that things are a little more complicated than we say. We're presumably turning on other proteins, which in fact then have the effect of reversing the on business. But anyhow, but at least we are modulating genes. And here's chromatin. Here's the histone in the middle. Here's a little tail of the histone coming out. And it's out here that the lysines are, whose specific acetylation of particular ones. All these lysines, of course, have been identified, and particular lysines have been identified as the ones that code for particular gene expression, gene transcription. And in addition, you've got this core in here. And then this is the DNA, of course, around it there. Now, what do we see on histone acetylation in these mouse? We're now back to the mouse cells for a minute here. What happens with SAHA on histone acetylation mouse cells? And this is really quite nice. It turns out if you don't treat with SAHA, this is what you see. These are the levels of acetylation you see with a given amount of protein. Kumasi stain is just a strain for levels of protein, and so that tells you we're not cheating. In other words, the same amount of protein is over here as is over there. That's what this is telling you. But you can see there's almost no acetylation of the histones in these cells. Now, normal cells have some. The first thing that one tells you right away is that abnormally low levels of acetylation may be part of the problem with cancer cells, because these cells have less than they ought to have. And when we upregulate it by putting in our histone, our SAHA, then we greatly increase the level of acetylation. You can put it in and wash it away in a while, and still 48 hours later, we still have some residual effect. This is called pulsatile treatment. You get a pulse in there, you turn on the mechanism by which this stuff is acetylated, you turn that on, and then you can get the drug away and it lasts for a while. That's going to be very critical to how this thing works and why it works as well as it does. Anyhow, you can see that we are acetylating all these things, and it hangs on for a little bit. Okay, an X-ray crystal structure was done by essentially Pavlitic, who's at the Sloan Kettering. Pavlitic is a great X-ray crystallographer, and he was trying to get crystals of the human histone deacetylase, but neither he nor anybody else could get it directly. Eventually, people have gotten some crystals of human histone deacetylase by chopping off some of the pieces at the end so that one could get decent crystals. But this thing is essentially a histone deacetylase-like. It's a homolog. It's called histone deacetylase-like protein, sort of a strange name for it. But it really has histone deacetylase activity. It's just not human histone deacetylase. And here's the X-ray crystal structure, and you can see here's our compound. Down here is the hydroxamic acid bound to the zinc. At the bottom of this cavity here, there's a lot of stuff here, which obviously I've erased. I just want to show you sort of the environment around the bound stuff. But here it is. It's going right up to this cavity, and up at the top here, there's a flat hydrophobic region, and the space between here and there is the 6 carbons. The magic of 6 is that's what it takes to get between the hydrophobic binding on the top and the hydroxamic at the bottom, right? So that's why it's the best. And if you have too much, there's some buckling around. And if you have too little, presumably some contraction of the protein can occur. at an energy price. I mean, if you have to pull that zinc and those things closer together, you can do that. You know, proteins are made of jello, right? They're not made of concrete, so you can do that stuff, but you have to pay the price of compressing it out of its normal structure. Anyhow, there it is. One little bit of story about this for the students in the audience, because you have to know the terrible realities of research, all right? Not the idealizations. Okay. Now, if you get a structure you really get an answer you really love, you have a great theory and you say, I know it's going to work, and by golly, it does. Your obligation is to test it as much as possible and try to prove you're wrong. Because better you prove it than the guy that you hate who lives in the other place there somewhere, you know? So you gotta be careful. You must test and try to prove that you're wrong. But suppose you get a result you don't like. What you don't have to say is, well, that takes care of that. Who knows that that's correct? And you ought to try at least to consider the possibility that was screwed up somehow. I got the message from the people who were looking at this. My colleagues had cloned and expressed this material. The people at Sloan Kettering had done this. And it had not been really characterized up to that point. And so I said, do a atomic absorption spectrum. I said, this is going to be metalloenzyme. I'm betting on zinc. Because you got a hydroxamic acid, and you're blocking things that are hydrolyzing peptides. And boy, that smells like a metal. Doesn't that mean all the metalloenzymes, that's what they do, especially with the hydroxamic acid as this particular kind of inhibitor. The carboxylic acid doesn't do it, for instance, okay? It takes that. So I said, then they called me up and they said, no, I'm sorry, we checked it carefully, and there's no metal of any kind in the enzyme, including there's no zinc. Now, my reaction is the one that I recommend to you students in a situation like this. I simply said, I don't believe it. Right? I said, I want you to check and make sure that you're right. And they called me back and they said, oh, we've tested it and all of the enzymatic activity is now gone. They had managed to purify the thing beautifully to the point where they got the zinc out of it. If you put the zinc in, it works. You put other metal ions, it doesn't. It is, in fact, a zinc enzyme. And so there's the zinc. So we put the zinc in there. And of course, the X-ray shows it. There it is, right? There's the zinc. It's a zinc enzyme. But we could have been way off the track if we hadn't been just as critical. Or maybe I was even more critical. Maybe I preferred to believe their result even less than I might prefer to believe my own. Anyhow, I said, I wanted you to just check and make sure that this is real. Anyhow, so it was real. It was not real. There is a zinc in there, and there it is. Anyhow, so that's that compound. And you can see why our stuff works. Here's a sort of a mimic up here of what the enzyme might look like, the protein. There's a piece of protein here, and then here's the amino group and the carbonyl group linked back to other parts of the protein. And here's the 1, 2, 3, 4, 5 carbons, 4 carbon side chain plus the alpha carbon plus the NH of lysine. And here's the acetyl group, and it's sitting right down here. And this is the thing we saw in the X-ray structure. We saw 1, 2, 3, 4, 5, 6 carbon, not nitrogen. Nitrogen doesn't work. It's interesting. I don't really know why, but anyhow, it doesn't. And therefore, the hydroxyurea, for instance, doesn't work well, but this one does. Methylene group there, so the same spacing. And here's the zinc, and you can see the zinc coordinated to the carbonyl oxygen. That's what you need to do this. This is what's called the zinc hydroxide mechanism. It's what carbonic anhydrase uses. It's sort of what carboxypeptidase uses. It's a zinc acting as an electrophile on the carbonyl while delivering nucleophilically a hydroxide to the carbon. And you can see that that and this look a lot alike. So what we had stumbled into, by the way I described it, without knowing what the target is, we had to stumble into a mimic for the transition state for this enzyme, right? So it's pretty good, and they're not so easy, therefore, for the enzyme to beat it. If the organism wanted to get rid of this, say the cancer cell wanted to get rid of this, it would have to figure out some way to get rid of it. Maybe it could hydrolyze this stuff. That could be hydrolyzed, perhaps. That would get rid of the problem. Or maybe it can excrete it. You'll see here, by the way, that I've shown you 2 different hydrophobic groups. The one I originally had showed you is the one over here linked to the carbonyl. Then we had 1, 2, 3, 4, I guess, no, it maybe it's this one here. It had a benzene ring here, the nitrogen, and then something down there. But yeah, we had that kind of structure before. But now, obviously, you look at this and you say, well, gee, maybe we need more hydrophobic stuff there. So let's do that. Let's get more active compounds. And so we made some much more active compounds. They were active at low nanomolar concentrations. Let's see, did I show you? No, I didn't show you the structures. Okay. They were the most active cytodifferentiating compounds known at that time. We patented the things in 1999. And here's an example. For instance, here's one that sort of looks like what I just showed you. Here's an amino group. This is an amino acid. The chirality matters. This has to be an L amino acid for it to work. It's just like the lysine. It has a lysine stereochemical preference. And you've got a carbonyl group on the nitrogen. Imagine that as the rest of your peptide chain, and something else hanging off here. Imagine the rest of the peptide chain down here. This was really potent. We had some evidence indicating that we could start suppressing the enzyme in the femtomolar region. I mean, it was really a highly potent compound. And this one was medium potent. This was single-digit nanomolar. And this one, you see, doesn't have the stereochemical problem. I mean, it's a symmetric compound here. But it still has the same kind of general other stuff on it for hydrophobic binding. These are both highly potent compounds. But highly potent compounds in this series are too toxic. And that is a very interesting finding that turned out to make a huge difference. It turns out scientists at Merck, these were people I knew very well. One of the leads of this guy had actually been a postdoc at Columbia one time with Clark Still. I knew these people, they knew about our stuff, and they said, this is a joke. These guys are making something that's micromolar, maybe high nanomolar. I mean, it's not, you know. For the women in the audience, let me tell you how men operate. You are not a real man unless you are at least single-digit nanomolar or sub-nanomolar, maybe picomolar. You know, it's a power thing, right? And whereas women are not dumb enough to do stuff like that, I believe. But anyhow, men are. And so what we do, the way people do this, many of the talks here of people in the business will tell you this is how you do it. You first make the most potent compound you can, then you test it to see whether it's toxic. It is toxic, you say, well, that takes care of that. And that's what they did. So Merck folded the program. But you'll see later that you can see already there's one problem with this theory. The theory is you can't make a drug in this series, but you know, there was one, right? We had one. So maybe that's telling you something about toxicity. And the answer is that high potency means slow off. Nobody knows how to make a drug go on faster. If you want to get a stronger binding, see, a binding constant is a rate constant on over a rate constant off, right? Ratio of these two rate constants, right? Nobody knows how to make them go on faster. If it's highly bound, it's because it comes off slowly, because when you finally get in there, all those binding interactions have been set and you have to throw them all away to get out of there. Off is first order. It's like radioactive decay. It neither knows nor cares what concentration in solution you're dealing with that got you your 50% or 90% inhibition. Therefore, what people think is that, well, make a highly potent compound, we'll just eat less. It doesn't work. Highly potent means that it hangs around. And in this series, that seems to be a problem. What's happening here is that we are modulating gene transcription. Well, I mean, lots of genes. You know, we're upregulating 300 genes and we downregulate another 300. We got 30,000, that's all right. So there's plenty of those left to go, but that's a lot of genes that we're modulating here. We knew this out of gene chips. So the answer here seems to be that what you need to do is get in there, get things started, and then get out before you are starting to cause serious trouble with modulating genes you don't want to, or maybe finding to pick up binding in weaker binding sites. People will say hydroxamic acids, they bind to every metal. Yeah, but not the ones that have this long chain and the hydrophobic at the end, and we need all that to get binding. You know, that's our specificity. But still, you think you better worry about getting the high potency compounds. Now, it's not always necessary to worry about that. If you have an antibiotic, you probably want it to hang around forever. But when you're playing around with gene transcription, you probably don't. And what we think is happening is that we are turning on the cytostasis. We know how. Let's get to that. We found out which genes are affected. Less than 1% are upregulated. That's a little less than 300. But one of the genes upregulated is P21. It's a known growth regulator. And then we have some other things that also were turned on. What happens, P21 is a material that freezes the cells in the G1 phase because it's an inhibitor of a kinase. Many of the talks about cancer today are about kinases. Kinases play a big role in cancer, right? It inhibits a kinase that the cell needs in order to go around the cell cycle. If you block that kinase, which P21 does, the cells are frozen in the G1 phase of the cell cycle. Our compound freezes the cells in the G1 phase of the cell cycle. We did this. We were able to find that stuff out. And if you suppress P21, you can release the SAHA inhibited cells from the G1 phase. In other words, we get cytostasis, but if we put an anti-P21, the cells start going around the cycle again. So it's really the P21 that accounts for the cytostasis, why the cancer cells don't keep proliferating, but just stay where they are. But we still get apoptosis. They start around the cell, and then next thing you know, they undergo apoptosis, so it doesn't really save them. There are a number of other effects of SAHA, and I don't want to go through all of this, but let me give you a reference right now. In the beginning of 2007, so this is a little old already, but this is sort of the last work we really did in this area. The business of 2007, in Nature Biotechnology, we have a review of this written by Paul Marks, who's my biological collaborator and me. And we had this thing where we summarize all the things that one knew about it. There are multiple cancer-related targets. I've given you sort of the simplest version of it. There are some special things that show up. For instance, you can modulate the proteins that are involved in cell motility, which is tubulin. That has something to do with spread of cancers. Okay, anyhow, apoptosis is programmed cell death, not general toxicity. So how does it occur? And what's the role of SAHA? Are caspases there's sort of two general kinds of apoptosis. One involving caspases, getting in right early, and the other one involves other things and then caspases later. And a collaborator, Astrid Roodflee in Australia, studies this stuff. So she took our compound and she studied it and discovered that the apoptosis that was involved here was characterized by cleavage of BID, which is a member of what's called the BCL2 family of proteins. It's regulated by P53. So some of you who heard some of the cancer talks today heard about P53. But anyhow, it's regulated by that, and you get a reactive oxygen species, either singlet oxygen or peroxide of some sort. I believe it's still not established which one it is. Anyhow, but the point is, that thing is to some extent worked out. What induces the cytodifferentiation? The most interesting part of this, what makes these cancer cells turn into normal cells? We don't have any idea. It's a very interesting biological question. We're modulating 600 genes. Somewhere in there is the answer to that, and people are working on it. But we haven't worked on it much because we were more interested in getting this into people and see whether it worked. Okay, in vivo animal studies. We put it in mice which had a human cancer in them. If the human cancer is allowed to grow untreated, after 20 days, the mice can't survive it anymore. If the mice have been treated with our compound, you can see with enough of the compound, there's no other problem. The mice tolerate it fine. There's no growth whatsoever of the human cancer in the mice. And with an intermediate amount, you can get some growth. So we can suppress the growth of human cancer in mice by using this compound. We also did some anti-cancer studies. These I'll try to go through fast because I think I'm going to get to the real crux of this whole thing shortly. This is a study by Lou Cohen at the American Health Institute in Valhalla, New York. And what he was studying was nitrosylmethylurea. It's a ferocious carcinogen, and it induces cancers in, let me get to this thing here. These are rats which are sensitive to this compound and get essentially breast cancer. They get breast cancer from this stuff. And so we looked at whether we could prevent that, and then Desai, in the same place, looked at whether this thing worked to prevent the lung cancer in these mice, which they get from this nitrosamine. I mean, the chemists in the audience will recognize this is a pretty simple structure with a very long name, but it's a nitrosamino-pyridylbutanone, so it's a ketone nitrosamine, and that's what you get in, that's in the tar of smoking that you get when you're a smoker, and it induces lung cancer in these females. But the results were pretty much the same for both. I'll just show you the mammary stuff here. Here, what happened, we had these rats and started feeding them an amount of the stuff which people thought was probably the right amount. We looked at a couple different levels, parts per million, 450, and then 900. They handled both of them just fine. And what we found is that when we just didn't give them the SAHA, and then you injected nitrosylmethylurea, 19 out of 20 got tumors, and the tumor volumes were really very large. Whereas if we gave them 900 parts per million in their diets, then only 12 out of 20 shows anything, and they showed very small tumor volume. So a decrease in the frequency and a decrease in the extent of the cancer. And the people, I mean, I would love to see zeros here, we don't have them, but the people at the Health Institute told me that this was the best chemo-prevented they'd ever seen, because these are ferociously nasty compounds that they're being treated with, and yet we're able to keep that under control. Okay, any tox? Mice, rats, rabbits, and dogs. No significant tox at effective doses. In dogs, this was really an amazing thing. There was continuous infusion in dogs over a number of weeks. At first, their white cell counts went down a bit, but then they came back to normal during the infusion. We weren't even permanently destroying white cells. We were simply suppressing them somewhat. So this was great. And in mice and feeding experiments, there were no physical or behavioral problems with these animals. So as a result of this, we got approval to do phase 1 clinical trials, which means get it into people. And then those were enormously successful. So then we got permission to do phase 2 trials. We've been in human cancer patients since March of 2000. So now you can see it's over 8 years already that's been in trials. And actually the trials were going on, well, the trials are still going on. I'll see something about that. But the trials that were needed for approval went on only for about 6 years or so. 200 human patients were looked at, over 200 human patients in the phase 1 trials. And we looked at breast, prostate, lung, Hodgkin's, non-Hodgkin's, bladder, many, many others. This is one of the sad stories, of course. You can always get people to take human trials because there's so many cancer patients around. And my collaborator at Sloan Kettering was in fact the head of Sloan. He had started off as the dean of our medical school. He became essentially the head of Sloan Kettering. And Memorial Hospital is one of the great cancer hospitals in the world. So we had, unfortunately, all the cancer patients you could want in these trials, all right? So anyhow, here, the results so far were very good oral availability. People swallowed this stuff. They got about 10 hours of duration. That's a combination in the pharmacokinetics. We're not sure exactly what it is. Some of it has to do with the drug getting compartmentalized and not completely hydrolyzed as fast as it could be. But in fact, we got 10-hour duration after a single dose of about 200 or 300 milligrams. So it's not some tiny little microgram thing, but it's about the size of what you take as a vitamin C pill. I mean, it's a perfect, you don't do it with a shovel, right? So it's a perfectly decent sized pill that you take, and this is what we do. It turns out that people handle it very well if it's done this way. Doctors also try doing it twice a day because then they can have it there all the time. And while in the dog there was no problem, people didn't feel that well. And so it went back to once a day. So this really means that what we get in there, we do what we have to, 10 hours later it's out of there, and 14 hours they have to recover anything that we started messing with that we didn't want to. And then the next day you hit it again, and that's plenty, because the effects seem to last on, as I showed you with this P21 story. We saw no significant human tox, even at high doses. More recently, a few blood dyscrasias have turned up, but not enough to worry about. But essentially, because other people have done more trials with our stuff. In our studies, we got all we saw was fatigue and some gastric upset or diarrhea, and this was all just handled with antacids and Imodium and things of that sort. So no big problem there. And so most of the patients showed useful responses. This is a number I really don't know in detail. I'll give you of the order of at least 30% of the patients were showing really good responses, and other people were showing some. And that is really good means evidence that the thing was disappearing, and cytostasis, that is static, it's stopping the growth of the cancer that we got with essentially everybody, but they didn't always disappear. Here's some examples. Here's a Hodgkin's lymphoma patient. This is a so-called CT scan, computerized tomography. You do a three-dimensional X-ray thing. And here you've got a big thing growing in the pleural cavity, another one here, another one there. Those are lymphomas. And then the patients, in this case, early stage, we didn't know. Nobody knew how much you could give the people. So they only gave it to them 5 days a week for 3 weeks. And this was the level of dose they gave them. And then they stopped. So after 3 weeks, they stopped. And then after 2 months from the beginning of the thing, they looked, and this thing had shrunk by 2/3. This thing had shrunk by 2/3. This thing is pretty much wiped out here. In other words, in spite of the fact that the drug was not in there all the time, we got processes started that essentially reversed the normal thing, which would say these would grow, and instead sent them backwards. That was also true here of a bladder cancer. Here's a big bladder cancer here again, 3 weeks treatment, and then it was stopped. And 2 months later, again, it was looked at. And it turns out that this large thing here had shrunk down to that little business there. There's about a 40% shrinkage instead of continuing to grow. So that means cytostasis plus some disappearance of the cancer rather than continuing to grow. Here's metastatic laryngeal cancer. This patient, the man had a cancer of the larynx, and it had metastasized into his pleural cavity. So these are the CT scans of his pleural cavity, the lung cavity. And you can see a big thing here, big thing there, big thing there, and there's even some stuff over here. And then after 8 weeks of treatment, now we didn't stop it at 3 weeks. It was clear people had no problem with it. We didn't stop it. Then, in fact, this is now completely wiped out. Those are dead cells. That hollow thing means those cells are gone. Up here, this big thing had almost disappeared. This business down here, essentially a tiny little trace of something down there, and even some stuff over here seems to have gone away. So you can see big disappearances of the cancers here. Here's another example. Again, a CT scan. You can see a big thing here, and after one cycle, which is a couple of weeks of the thing, in fact, it had gone from there all the way down to this little thing here. Here's a gastrohepatic mass, big mass, which in fact, after one cycle again of the drug, it completely disappeared. It was complete resolution of that mass in this stuff by just swallowing this pill, as I said. Now, here we get into some cases where you can actually see not just CT scans, but actual cancers themselves. These are patients who have what's called cutaneous T-cell lymphoma, and cutaneous, obviously, on the skin. T-cell lymphoma, it's like any T-cell. There are two kinds of lymphoma, T-cell and B-cell. This is T-cell lymphoma, and it's expressed on the surface of the skin, so you have this big business here, and in addition, you have these things on their arms. You have these things here and those things there. Those are all expressions of that same disease. After 1 week of SAHA, this is almost clear. You can see there's a little bit of stuff here, but it's almost clear. The arm is clear here. This arm is clear. That is because, besides this being, in fact, a progressive and eventually lethal cancer, it causes tremendous itching. I mean, being on the skin like that, it really drives them absolutely crazy. And this guy was digging away at his arm. There's a big thing up here that he did, and they just bandaged that up. So that's not hiding some terrible story about our drug. That's simply, that's what happened. Now here's the back of the guy. You can see it even more clearly. The back of his legs. You have all this stuff. The same guy. After 1 week of SAHA, the same guy goes from that to this, where essentially it's all gone. It's all gone now. And so that's pretty good. Now the next one is hard to take. We're going to have a barbecue, and I was told that there isn't enough food for everybody. And so I'm going to show you this next slide, and it'll kill the appetite of most of you, right? Solving the problem. Here she is. Here's a woman who has cutaneous T-cell lymphoma on her face. She's got a big thing like this, another thing on top of it. The things over her eyes are absolutely awful. She, in addition, has all these bumps down her neck. She's got stuff in her ears. And now, after 8 weeks of SAHA, she goes from that to this. A little bit of scarring here. So that one is not bad, but this one's pretty hard to take. A little bit of scarring here where that was, but fundamentally it's clear. The thing over her eyes is gone. The thing over her neck is gone. It's really great. Great results, you know. If this is the best we ever do, I mean, think of what we did for this woman. Unbelievable. And here's the top of her head before. You can see a big bump here and a lot of these lumps down her neck. And afterwards, again, all that stuff there, all that stuff is cleared up. And the top of her neck, the top of her head, she has a little bit of scarring here, but that's pretty much it. Now, what you can see in addition, this was only after 8 weeks. That was right away. What I've heard in addition to that, it turns out she originally, of course, as a woman, she had a perfectly nice head of hair, but this disease knocked off all her hair. It wasn't the drug. She had no hair left as a result of this disease. And here you don't see it yet, but actually a couple months later, her hair is back and it's normal. So people have often asked me, what is the worst side effect that you see with the drug? And we say the worst side effect that we can't overcome is it seems to grow hair on bald heads. But maybe not. Maybe only if you lose your hair because of this disease, right? Okay, so combination therapy. With excellent results in cancer cells in combination with some leading anti-cancer compounds. So far, it's been looked at ones where the cancer compound works because you go around the cell. You know, if you have to go around the cell cycle, it won't work. Let's put it that way. We haven't really checked that yet, but it makes no sense that it would if we're freezing it. But gemcitabine is a drug that doesn't require that. We get great combination, and I'll get to that in another. This is the one that's really interesting to me. There's a man who studies how much X-radiation you need to kill cancer cells. So he treated the cells with our compound, and it cut way back on the amount of radiation you needed to do. That means that really people getting radiation should be taking this kind of thing and cutting back on their doses and getting a better result. Yeah? Anyhow, we had a meeting with the FDA. We showed them things like the picture of this poor woman and things of that sort. They said, okay, you can go to phase 3 trials. They gave you fast track. Fast track means we would act on it within 6 months. Orphan drug, that means we were hitting a disease that other things didn't hit, and therefore you get some special protection against competition while you have this orphan drug category. Anyhow, so we got that, and in the meantime, we had founded a small company. We had founded a little company. We founded it in 2001. You see, I said we were in humans since 2000. Early stages of human trials since 2000 were funded with just money we dug up somewhere. But then eventually for phase 2, we really had to have serious money. I mean, it costs at least $10,000 a patient to do this kind of stuff, and that was a lot of money. So we founded a little company. We looked at selling shares in Columbia University and Sloan Kettering, but nobody was crazy enough to buy those shares. And so we then had to find a company. So we started a small company. Columbia and Sloan Kettering got shares of the company in return for giving us the rights to the patents, because those of you in the academic business know that patents belong to the university, but they gave us the rights to that in this little company. So we founded this company in 2001, and Merck bought this little company, lock, stock, and barrel, everything, in 2004. And for good reason, I think, because the truth is they finally realized that looking only at the most potent compounds was probably a mistake, that the medium potency compounds is what they should have been looking at, because that's what we had. And so they bought the whole thing, including all our patents. And the phase 3 trials for this cutaneous T-cell lymphoma were successfully completed very shortly thereafter. We had about 6 months after that, they filed then, and they filed for approval in June of 2006. And we got approval in October of 2006. It was actually less than 4 months. Now, it's my own opinion, based on extensive experience, that the government can't do anything in less than 4 months. So they must have started typing the approval as soon as they got this stuff in order to get this out. But they did approve it in less than 4 months. And in fact, it was approved then in October of 2006. Merck renamed it Vorinostat, the generic, and Zolinza. If you type SAHA into Google, you'll see there are hundreds of things that are called SAHA. You couldn't use that. So they had to come up with these names, wonderfully euphonious names, right? Beautiful names. Anyway, so that's what they did. So that's what the things are now called, and Merck is now handling the whole thing. We have clinical trials going on like mad. More than 72 clinical trials going on in March 2008. You can see that there's a website called clinicaltrials.gov, which lists all the clinical trials against any particular disease. So for cancer and for Vorinostat or SAHA, they still recognize SAHA. We have trials going on, brain cancers. This one we're getting really good results. And as I say, you're going to hear from Laura Kaufman if you go to her talk tomorrow about what they're finding in the laboratory. Mesothelioma, that's the asbestos-induced lung cancer, that's very far along. And the results are really great. And a lot of other things here. We're doing combination therapies. We're finally doing combination with radiation, an important thing to do, because as I say, I think that's a great thing. So a lot of clinical trials are going on with the compound, and it looks really very good. The people who are involved in this, Paul Mark, Richard Rifkin, Victoria Richon, and Kevin Kelly, these are the people at Sloan Kettering. Paul has been my collaborator forever, ever since he came down and told me about these findings of Charlotte Friend. Richard Rifkin, he was the dean of our medical school. Richard was in the Department of Medicine there. Paul became director of Sloan Kettering, and Dick came along as the director of research at Sloan Kettering. So I had that institute more or less focusing on us. Vicki Richon was a biochemist working in the lab who did a lot of really lovely studies of the kinds I've referred to here. Kevin Kelly is a physician at Memorial Hospital who ran the clinical trials at the beginning. Eventually, the clinical trials are being done everywhere. They're all over the country, but they started at least at Memorial Hospital. My people, I had so many people in it, I couldn't possibly list them. I ran this to some extent as a lottery. I said, everybody who has a project, make a couple compounds, throw them in, see if you're a winner, you know? So everybody had their own version of it. But these are some of the people who did interesting things. Yael Webb really worked out the whole mechanism by which the thing works. Sandro Belvedere made our most potent compound, and then that was how we learned that high potency was not acceptable in this series. Leland Gershel was both an MD and a PhD with me, and he in fact then is now off with his own little company somewhere. And Tom Miller became the director he was my postdoc, he became director of chemistry in our little company, Aton. And then when Merck bought us, then Merck hired him as well, and he's now running the project actually at the Merck labs in Boston. And Vicki Richon was our biologist. She became in my little company the biology director, and then Merck has also hired her, and she's doing the biology part. The other thing I can tell you about this without doing anything more, because I realize I'm pushing my luck here with the barbecue already in full blast here. The other thing I can tell you is that we're modulating gene transcription. Now, that's interesting for cancer, but it surely can't be the only place where that would be interesting. And it isn't. We have, according to the person, there's a woman named Nancy Wexler, who's the head of what's called the Huntington's Chorea Foundation. Nancy and I were talking about this, and she decided she should try our compound in her animal model. And she did. And it turns out she told me about a month ago that this is the only compound that works in the animal model to stop the progression of Huntington's disease in the animal model. It's not yet in human trials. Why? I can't imagine. Because that's a dreadful disease for which there really isn't anything yet. And now that looks as if this might be useful there too. So I think what we've done, besides whatever we managed to do with some cancer business here, I think we've opened up two things. An approach to dealing with many diseases which involves modulating gene transcription rather than just poisoning things, right? That's a good thing to do. And in addition to that, many, many places where it can be applied. So, and many places where people are trying it. I don't know of any drug company that doesn't have somebody chasing us, trying to beat us on our patents, and they may succeed. I would say we're the first. We're certainly not going to be the last. There are going to be plenty of people doing this, and that's fine. And if the result of that is even better cures for people, and even less money for Columbia University and for me, and things like that's all right. Anyhow, I thought you would find it interesting to see this story because it started with such a ridiculous DMSO and built onto something that really turns out to be pretty good. So thanks very much for your attention.
Thank you very much, Dr. Breslow. If there are any questions.
Let's see, what do you want? You want this thing here? Let's see.
I have two questions. Do patients continually have to receive the drug, or are there courses of treatment like many anti-cancer drugs? That wasn't clear. And the second question I have is that the hydroxamic acid moiety is used in things like betimastat, which have also been used against matrix metalloproteinases, and I just wondered whether they could also be targets for SAHA.
Yeah, I think we don't know yet about the time course of treatment. And as you know, we only got approval in 2006. Well, that sounds like a long time ago. It's been a year and a half. I think that's probably not long enough yet to know about how long you have to do this. Do you get a recurrence if you stop? Do you get a recurrence if you don't stop? I mean, there are any number of serious questions here, obviously. So far, I haven't heard of any problems of that sort, but I think it's too early to know that. And do we have any side effects of long-term use of the drug? Do we need to use it long-term? The question you asked. Do we have to use it long-term, or can we terminate it for a while and then see what happens? And I think that was done with many of these people, of course. It was terminated to see then what happens. But I'm not sure what happens after that. It may be that all we're doing is buying time. I don't know that. I know that in the glioblastoma trials, I've looked at the data that they have so far, and what they see is prolongation of life, but it's about doubling. In other words, the normal life expectancy isn't much, and they get about twice what the normal would be. Well, that sounds like a good number, but 2 is not the same as 100, So I'm not sure that we yet know what the potential is for this treatment and all the other things. And I didn't understand the second part. What was the second question? Yeah, right. Yeah, we worried about matrix metalloproteins because obviously many of them are zincs or whatever, and you're putting in hydroxamic acid, you're going to land on it. See, what we have here is a compound whose potency is only of the order of micromolar or maybe a little bit better, And much of that is contributed to the side chain, not to the hydroxamic acid. If you didn't have that 6-carbon business, it isn't any good. If you didn't have the hydrocarbon at the end, it isn't really any good. So I think what we see when we look at matrix metalloproteins is with the levels that we use, there is nothing, because they don't have the rest of the recognition site. But it's conceivable that if we use very large amounts of it, we would start hitting them too. You're not going to hit P450. P450 is a heme, and you can't hit heme because you got to chelate it, and you can't chelate an iron when it's got a porphyrin around it. So we don't have to worry about heme. But matrix metalloproteins, for sure, we looked at that right away, and that's important to know. Does it turn out that there is some problem? Well, don't forget, we give it for a while, and then it stops. That may be helpful with we don't know where else. With the dogs, we didn't see any need to stop. Nothing bad happened to them in continuous infusion. But with people, we do find they don't feel as good with twice a day, and they don't seem to need it, so we don't do that. So you got 14 hours with the drug not there. That may cure a lot of ills. I think there's a possibility that that may be an important thing to do. And so that's why, in a way, what you want is a compound that has the Goldilocks formula. Remember her Goldilocks? Not too hot, not too cold, just right. Remember the cereal she was trying to eat there? Well, what you want is the same thing in a drug. You don't want it so hot that it's so potent that it hangs around and either hits other targets, maybe not as well, but still well enough to cause a problem, or hangs around with your real target long enough that it gets you trouble, you know? You don't want that, so you really want to get in there and get out. In this case, provided what you're getting is pulsatile treatment, where a pulse is enough to start the cytostasis. It turns out P21 hangs around for a while, so we know that. Start apoptosis, you're not going to be able to reverse that. Once these cells start down that pathway, there's no evidence they can suddenly change their mind and go back again. I mean, so that probably is an irreversible process. Cytodifferentiation, we just don't understand. But maybe that's pulsatile too, you know? Maybe it is, and maybe that's why this whole thing can be done by getting it in, getting it out, not having it so potent that it hangs around. Think of a compound that we now get 10-hour lifetime. Think of a compound 100 times more potent than ours, and imagine that you get that 100 times by changing the off rate. So the thing hangs around 100 times longer than this 10 hours. It's there for 1,000 hours. I think you could have real trouble at that point. That's why you need a compound of this kind of potency to get in there and do its job and get out. And that may be the answer to the metalloproteinase as well. It may be that just getting it in, getting it out. If there is any problem with sequestering some of the metals or it binding to them as well, maybe you can get off again, you know?
You've convinced me that medium potency might be better than higher potency, but you know, I am a male, and just like sports cars, they may not be practical, but they're nice. And so high potency is still fascinating from a chemical point of view. What was necessary to get the highest potency, the slowest off rates for these compounds?
Well, I mean, I did something there, which I'll get away. What happened, of course, is that when we went to the one compound that we made, which was really sub-picomolar. It was at least was low picomolar and maybe sub. I mean, it seemed to be femtomolar. What we saw there is that the animals did not survive. Now, I don't know what killed them. And that would be an interesting thing to know, and maybe some of my colleagues know it. But when we do this stuff, those compounds were just fine with the cells. They weren't killing the cells, but they were killing the animals. And so I think that, you know, the mice, the rats, you know, the first animals, they were killing them. I don't know how they did it. That would be something worth knowing. If we understood that, maybe we could figure out how to get high potency and avoid that problem. And so there are many, many biological questions unanswered in this thing, and we purposely did that. My reaction is, let's get this thing to the point where if it really works, let's get to the point where we get it out and get people under treatment with it. And then at our leisure, assuming that we all then live forever, we have plenty of time to work out how it happens, you know? Once we get rid of the death from this thing. Yeah. Was that it? I have a feeling that was it. Oh, there's a question back there.
Is it possible that the stochastic behavior of gene expression is also complicating the time dependence you're so concerned with?
The stochastic behavior of?
It's the fact that the gene expression is occurring in a probabilistic manner in a cell.
Yeah.
And you have very different time dependences. The binding constant is not the only significant matter. It's how the probabilities of the distributions are concerned, given genes.
Given well, I mean, it's an interesting question. How long does this stuff have to be around in order to modulate the gene transcription to the point where we get the effect, which is supposed to bring on? I think we don't know that. What we know is two things only. At modest concentrations, we get all the effects that I showed you. People that weren't taking huge amounts beyond what I told you there. At modest concentrations, we get all those effects, and when we look at it, there are at least 2 out of the 3 effects that we know are affected by pulsing, and then they hang on. That is, P21 hangs on for a while even after we get the drug out of there, and apoptosis surely doesn't, the things don't suddenly turn around and start back again once they're on their pathway. I think that's pretty likely to be true. The side of differentiation, we just don't know. But we don't really know how long things have to be there to do their biology. But maybe what we can say is that one shot isn't enough. People do take it the 10-hour, they take it once a day. They take it once a day, as you saw, for several weeks. Sometimes they got results right away. Sometimes it took longer. There are things that are taking time on the biological scale, but the main thing is we are not keeping the drug there all the time, and it doesn't seem to be needed, and that seems to be a problem. But we don't know how. I don't know why you have to give it to people for 8 weeks before the cancer all disappears. Could you give it for 1 week and just stand back and watch? You saw several cases where that seemed to be true. We gave the thing for 3 weeks, and then looked at it 2 months later, and the cancers were still almost gone. Now, I don't think they happened after 3 weeks, but I think they were continuing to degrade essentially.
Well,
There's a barbecue out there, and you probably have recovered from that awful picture I showed you, and so you can go out and try to eat some of it.